So are there any questions from what we talked about last time before we get started? So there are pictures that I did put on there on Canvas for you that go with this section in here, and I have the pictures up for 6. I wanted to get started on 7 yesterday and just ran out of time for doing that. But when there are some that I feel like are really essential for you to print out and bring to class, I'll post on there. Bring this one. I will highlight it in the list in there that those are ones you're going to definitely want to have. When we get to the metabolism and photosynthesis, that's where you'll want to have them the most, because they're just long chemical pathways that you can print out instead of having to write down in there. So in this section, we look at the nucleic acids, which is the stuff that your genetic material is made out of. And the genes are going to be your unit of inheritance. They are made of DNA, and they're going to determine the primary sequence of the polypeptide. But the DNA is much bigger than the genes. So when we look at your DNA, you've got a long line of DNA. The genes are only about 1.5% of it in humans. In some animals, there are a lot more of it. In prokaryotes, they are almost all of it, but in humans, it is only 1.5%. So the DNA is the deoxyribonucleic acid, where RNA is ribonucleic acid. So there's just slight differences in the structure. In there, you have oxygen difference in there. They're both made up of a ribose sugar in there, which is where the ribo comes from in the ribonucleic acid in there. And then your chromosome is going to be the long strand of DNA that's going to cover, in most cases, over 100 genes. The Y chromosome in males right now, the thought is it's got about 78 chromosomes, but that number may change, or 78 genes on it, but that number may change as well in the future. For a while they thought it only had about 30. But that's the smallest one. In humans, they're usually going to be much bigger than that. So the chromosome is that one that when you picture in the cell, the little X-shaped thing in there, that's going to be the DNA that's all coiled up into an organized form. So your mRNA is the messenger RNA. When we make proteins in there, this is going to be the one that carries the message out to the workers in the cells for doing that. It's going to work with the synthesizing machinery in there so that you can use the DNA to make an RNA and that RNA to make a protein. And your ribosome is going to be the organelle that does the protein synthesis. We will go into that in more detail. And I've actually got a little activity that I made with these magnets that you can kind of go through and put the pieces together like a puzzle and watch them go through like that. That if that helps you to learn it too, I can give you the template that I made the magnets out of, and you can print them out on paper at home to use them as well. So a polynucleotide is a polymer of nucleic acid macromolecules. And that's not a term you'll see used that often. Polynucleotide just sounds kind of awkward in there. So we have nucleotides and nucleosides. A nucleotide has 3 parts: a nitrogenous base, the 5-carbon sugar, which is a pentose sugar, the pent for 5, and a phosphate. The nucleoside is the pentose and the nitrogenous base without the phosphate on there. And I do not have a quick little mnemonic for helping to remember the difference of it. Wow, this thing is just incredibly sensitive this morning. It's doing things all on its own. So the families of nitrogenous bases, we have the pyrimidines and the purines. The pyrimidines are going to be 6-membered rings of carbon and nitrogen, which is what all the bases are made up, because they are going to be made of amino acids in there. They are considered bases because they will take up hydrogen ions from a solution. The ones that are pyrimidines are cytosine, thymine, and uracil that we abbreviate as C, T, and U. The purines are bigger. Those are going to be a 6-membered ring fused to a 5-membered ring, and they include adenine and guanine, or abbreviated as A and G. So the pyrimidines they have the longer name but the shorter structure. The purines have the shorter name but the longer structure in there. So ribose is a 5-carbon sugar that's in the RNA. And deoxyribose is going to be a 5-carbon sugar lacking in oxygen on the 2nd carbon in the ring. So that's why it's called deoxyribose. And you'll hear them talk about DNA having a sugar phosphate backbone, and that's what we mean with the sugar phosphate in there. So your atoms in the nitrogenous bases and sugars are numbered so that the carbon atoms have a prime after the number to distinguish them. So when we talk about this, the 5' carbon in there, that way we can tell which DNA end of the DNA is which. So when you have the 5' at one end, the other one we look at is 3' at the other end. So what's going to join all of these together is a phosphodiester linkage, which I know that doesn't mean anything to you right now. It's just going to be the type of bond that joins those adjacent nucleotides in there. So you've got a phosphate group that links to 2 sugars. So you've got sugar, phosphate, sugar in there. So your backbone, which is like the sides of the ladder, is going to be a repeating pattern of sugar, phosphate, sugar, phosphate, sugar, phosphate. But what is actually on the ends is different. One end is going to have a sugar, one end is going to have a phosphate. And so your 5' end is going to be the one that has your phosphate attached to carbon number 5. And then your 3' end has an hydroxyl group attached to carbon number 3. So when we look at this, you're going to have your 5' end here. And your 3' end at the other end. The reason that's important is because when we read DNA, we read it from the 5' to the 3' end on there. And you just want to get that idea in your head and have it be a little phrase that you've heard a lot, 5' to 3'. And that's the easiest way to remember it in there. So DNA builds from 5' to 3'. And so what's going to happen is you go down and you're reading in here, this area that's the gene is going to be what is going to code for the specific sequence of the 4 DNA bases, and that's going to determine your primary protein in there. So that's going to give you your amino acids that make up that primary string in there. So DNA is shaped in a double helix. It looks like a ladder that somebody has taken it and twisted it. So if you just picture it being twisted like that. And this is going to be your sugar phosphate backbone on the sides in there. So RNA in most cells, it's a little different in viruses, but in most cells is going to be just your single strand in there. DNA will usually be double-stranded. And the arrangement is anti-parallel. So when you've got the strand here, this one goes 5' to 3'. The complementary strand runs in the opposite direction. So if you think about it, is you've got phosphate, sugar, phosphate, sugar, phosphate, sugar, going 5' to 3'. The other way, you're going to have phosphate, sugar, phosphate, sugar. And then you've got those base pairs that are in between connected to it. So the sugar is the one that has the hydroxyl group on carbon number 3. The hydroxyl is the OH. So when you look at names of things, when you see an OL ending on it, it's telling you it's an alcohol. So some of the sugars are sugar alcohols, so like xylitol is a sugar alcohol in there. So the way the bases are going to pair on here is going to be in a complementary nature. So it's kind of like a lock and key mechanism. In DNA, you're going to have A and T paired together. And G and C paired together. I'm sorry this gets kind of hard to see for you guys over there in the corner. My leash is short. There's no pictures on the test. You will want to know how they pair, but this kind of helps you get the idea that it's complementary. So by being complementary, it means that it doesn't pair with its exact match. It pairs with one that fits like a lock and a key. You can't put this key in this lock. You can't put this key in this lock either. They only go with their own in there. So in DNA, it's going to pair that way. In RNA, you don't have thymine. You're going to have uracil instead. So adenine is going to pair with uracil, and guanine and cytosine are still the same. Exactly. So if you have a strand of things that are like this, you can predict what goes on the other side. And the way we write that strand out is we would write it as so if you see letters like this, A, A, T, C, C, G, you can predict on this end you're going to have T, T, A, G, G, C. So that idea is critical to how DNA works, because when it's reading the code, your amino acids are actually going to have little labels on them that are going to match up to that, so that you know you're bringing the right amino acids in there. So what's going to hold things together in there, you've got hydrogen bonds that are going to hold these bases together. And you also have those van der Waals interactions. Those are the ones that are like the little hairs that allow spiders and lizards to walk on the walls. Those are going to help hold the DNA together, the two strands. So DNA has been important in studying evolution. There's molecular genealogy. What we expect is that two species that appear closely related on the fossils and anatomical evidence to share a greater portion of the DNA too. And for the most part, we found a lot of those things to correlate like that. So their protein sequences are going to be more alike than more of them that are distant relatives. And we also see that within an individual species and an individual family. Your DNA is going to be more like your family members than it is to somebody else in there. So also with your siblings in there. The similarities in the DNA, some animals have more variation in there, some have less. Humans have relatively little variation in our DNA amongst the differences between us. So when you look, we all look at each other and we say, well, clearly everybody in here looks different. It doesn't take a whole lot of variation to create something that looks different in there. DNA is the whole big long sequence, and the gene is the part of that sequence that actually tells you what protein to make. So it's a small portion of it. Yes. So what may code for blue eyes may only have one different base pair than what codes for brown eyes in there. It's the genes are, we can look at it and say we know that that gene is on this location in the chromosome, and that you're either going to have this variation of the gene or that variation of the gene. The variation of the gene that you get depends on what was passed on to you from your parents.
So, like, when somebody pops up in the family, like, and my brother has bright red hair, and nobody as far back as we know in any part of our family has red hair. Like, is that just, can that happen without somebody having that, or does that?
There are recessive genes out there that if you have two different genes, because you inherit one from mom and one from dad, one is going to be a dominant one and one's going to be a recessive one in a lot of cases. So the dominant one is going to show in there. So like with me, everybody all the way back on both sides of the family has blue eyes. I am the only one on both sides of the family that has green eyes. Sometimes you get little mutations and variations that occur in there. So my mom also had numerous x-rays. That's how they found me when she was pregnant, is on an x-ray. So, you know, maybe that had something to do with it. It may explain a lot of things about me, but occasionally you do get variations like that because it will happen with blood types as well. Those are actually codominant, so you inherit part of it from mom, part of it from dad in there. And there is possible combinations that would come. If you've got a parent that is a type A and a parent that's a type O, you can make a type A or a type O child out of that combination. That's it. And then all of a sudden maybe a type B baby pops out of there. Yes, you should be asking questions, but it is possible that a mutation could have occurred and it wasn't a Jerry Springer type of event that led to it. So, but they can do genetic testing that's going to be much more accurate for determining paternity in there than that. So that's going to be the end of the material that will be for the next test. And